Hi, so this will be our second lecture covering the material of chemical bonds, acids and bases, and what pH is, and then we will take a quick overview of all of the biological macromolecules. So what I'd like to start out with is a quick look at sodium. So this is a refresher from last time. So if we're looking at a sodium ion, which is pictured on the sodium atom, which is pictured on the left, and it loses an electron, how does that impact the charge of sodium? So I'll give you about 5 seconds to think about that, or you can pause. But okay, we're going to go to the answer. The answer is the charge of sodium is now +1 because it has lost one negatively charged electron. So ask me in class if you have any questions about why that's true. What I'd like to do next, or what we're going to do next, is talk about chemical bonds. So first bond that we're going to talk about are covalent bonds. Covalent bonds are ones in which the outer electron, so the valence electron, electrons are shared. So if you remember looking at an atom, so if it's nucleus, here's the inner shell, this would be the outer shell, so that would be a valence electron. So if you share a valence electrons between atoms, which you can see here in this bottom picture between two hydrogens, they're sharing the electrons there, which gives it 2 electrons in that one shell, and that shell makes it very stable, and this creates a very strong bond. It's actually the strongest of the chemical bonds. The next type of bond you find in human physiology are called ionic bonds. Ionic bonds are the bonds that are created when you have a cation and an anion attract. So cation and anion attracted to each other. And what happens when this happens is one atom donates the electron to the other. Donates electron to other, which you can see down here. Sodium is donating the electron to chloride, and so then that gives us a stable molecule, which then forms sodium chloride crystals, which is what we think of as salt. What happens is that these bonds tend to break up and dissolve when they get in the presence of something more attractive, such as a water molecule. So it's a weak bond. Breaks when something more attractive is present. Which you can see here. So here sodium chloride is dissolved in water, and the individual chloride ions and sodium ions have been broken apart and are now interacting with the water molecules. Okay, the third type of bond that we're going to talk about are hydrogen bonds. And these are bonds that are created when the slightly positive hydrogen atom binds to a weakly negative partner. So slightly positive hydrogen, Trojan atom binds to weakly negative partner. So in here we've got the positive charge of the hydrogen atom, the negative charge of the oxygen atom, and so then you get this interaction, which is the hydrogen bond. This is a very weak attraction, so even weaker than the ionic. So there is another type of bond, but because it's not terribly significant for human biology, we won't be talking about it, the van der Waals forces. So that, we're ready to move on and start talking about pH, acids, and bases. So pH is a measure of hydrogen ion concentration in solution, and how you calculate it is, I'm going to change colors to blue. So pH is equal to the negative log of the concentration of protons or hydrogen ions. And so what this tells us, so the negative inverts the scale, meaning that as the concentration of hydrogen ions goes up, pH goes down. Log tells us that this is a log scale, so that for every one-fold change in pH, you get a 10-fold change in hydrogen ions, which is what I wrote right here. So basically, as pH goes up, concentration of hydrogen ions goes down. So there are two types of proton acceptors and donors. An acid is a molecule, or primarily molecules or atoms, that donates protons. So proton donor. And proton is just another way of saying hydrogen ion. And a base is any sort of molecule that accepts protons. And the reason this is true, or why they're called this, so an acid donates protons, so it's releasing protons into solution typically. Which then means that as you increase the concentration of protons, pH goes down. And proton acceptors bind up those, so sponge up protons. And so pH goes up. So typical pH scale, which is down here, is acidic, very high concentrations of protons, pH 1. So high proton concentration. Whereas you can go to the other extreme, a very basic or alkaline solution, where the pH of 14, and you have almost no protons present in that solution. So, quick question. What is the concentration of hydrogen ions in a solution at pH 5 relative to water, which is pH 7? So I'll give you a few seconds, or 5 seconds, to think about this. This gives you a chance to pause if you'd like to sort of think about it more in depth. Okay, and then moving to the next, the answer. So the answer is, as you go from pH 5 to water, or as you go from water to pH 5, because this is going to be a log scale, we're going 2 pHs lower. So 2 times, or 10 times 10, let me erase that, I'll just cross it out. 10 times 10 equals 100, and so that's a 100-fold increase in hydrogen ions. Okay, and with that, we are done with pH, we are done with chemical bonds, so we're going to talk about the large organic molecules. So this is the field of organic chemistry. Organic chemistry is basically the study of this atom here, which is a carbon atom, and how it interacts with other molecules. There are basically 4 categories of carbon compound. We'll talk about each of these. So to start out talking about carbohydrates, we'll then talk about lipids, then proteins, and finally finish up talking about nucleic acid. But before we start this review of organic molecules, what we're going to talk about are uh, what's the commonality between all of them. So all macromolecules are polymers, which just means that they're a string of monomeric units. So a string of monomers. And they are synthesized through this process of dehydration synthesis, which means that as you combine monomers, you release a water molecule, which you can see here and also illustrated over here. So as this monomer and this monomer bond together, dehydration synthesis causes a release of a water molecule. So the converse reaction is called the hydrolysis reaction, and this breaks apart polymers. So it breaks apart polymers. And to do this, it requires a water molecule become incorporated into those two separated molecules. So it requires the atoms from a water molecule. So a way to think of this is dehydration synthesis is basically you're dehydrating the molecule, releasing water from it to synthesize it. Hydrolysis, so lysis is breaking something apart. You're breaking it apart with water, which is hydro. So hydrolysis is break apart of a molecule using water. So first of the organic molecules that we'll talk about are carbohydrates. So carbohydrates are molecules that are quickly metabolized sources of energy, and what the body does is convert most of these to glucose, and then oxidizes the glucose molecule to produce ATP, or adenosine triphosphate, which is then used to power all of metabolism. So basic chemical formula for carbohydrates is one carbon molecule for every water molecule. So one carbon for every one water. And then you can do this up to almost an infinite number of molecules. So N is the number of carbons that are involved in this. And the picture I have here are all from Voodoo Donuts. For those of you that are from Portland or not from Portland, this is probably the most well-known donut shop in Portland. I don't think it's the best, especially when they have these squishy, I don't even remember, it's like Lucky Charms donut, which I remember thinking looked good, but no. Same thing with the bacon one. There's better donut shops. Ask me if you're interested. So monosaccharide is the simplest of the carbohydrates. So this is a single sugary subunit, and it's produced by the digestion of more complex carbohydrates. The primary carbohydrate or monosaccharide that we find in our blood is glucose. So when we talk about metabolism and production of ATP, people typically talk about glucose. I will not be asking you to identify these other ones, but these are just some examples. So this is glucose here, and you can see there's some slight variations in some of the side chains, basically flipping this hydroxyl group in the proton to give you galactose, or changing some of the other side chains, adding in an extra carbon to give you fructose. But again, I won't be asking you that. What I want you to know is that a monosaccharide is a monomer of sugar, and that glucose is the main one that our body uses. We can then get into disaccharides, which are two monosaccharides linked together. So you can see they're joined together by a bond. And some common ones that we encounter, So probably the primary dietary form of sugar is sucrose, which is combined from one glucose. So here's the glucose and one fructose. Whereas lactose, which is what you find in milk, including human milk, is made up of a glucose and a galactose. Or maltose, which is the form of sugar that's most common in grains, is made from a glucose and a glucose. Okay, these molecules are actually joined together, because they're biological molecules by dehydration synthesis. And this bond that I circled here is a glycosidic bond. I'm going to go back. So next level are polysaccharides, and these are long chains of monosaccharides. And so here you can see monosaccharides, so it's a glucose, all bonded together. As you get to some common polysaccharides, probably the most common one in animals, including humans, is glycogen. Glycogen is the energy storage polysaccharide found in animals. So energy storage in animals, of which humans are one. And glycogen is synthesized primarily in the liver, but it's also synthesized by muscle cells and neurons, or support cells in the brain. So by liver, muscles, and brain. When we eat animals, this tends not to be the primary source of energy that we ingest, but is kind of when we store energy in the form of a polysaccharide, we use glycogen. Starch is actually one of our primary sources of carbohydrate, and this is the form of energy storage found in plants. And it is the only significant digestible polysaccharide in our diet. Dietary polysaccharide. Cellulose is another form of polysaccharide that's not digestible, so even though it's in our diet, so when you eat lettuce, there's a lot of cellulose in there as well, but we're unable to digest it, and so it just passes through our bodies. Okay, with that, we're ready to talk about lipids. There are three categories of lipids, or first, general properties of lipids. Lipids of this group are the only ones that do not truly form polymers, so they're sort of an outgroup of the large biomolecules. They tend not to be terribly big, and they're also very hydrophobic, meaning that they do not like water. So phobic for fear, hydro, water. There's three primary categories. We'll talk through each of them: fats, phospholipids, and then cholesterol and cholesterol-derived molecules. First, we'll talk about triglycerides. So triglyceride is basically a glycerol, which is this chain over here. Glycerol plus 3 fatty acids. So hence the name triglyceride. So glycerol and then tri for the fatty acids. When we talk about triglycerides, these are one of the primary dietary sources of energy as well as storage in our body. There's two forms. There's saturated fatty fats and unsaturated fats. Saturated fat is one in which there are no double bonds between the carbons. No double bonds. Oops, my pen is not working so well. Double bonds. And so what happens with saturated fats is that they are solid at room temperature. which is not terribly healthy for us because it can also solidify in our blood and occlude or block arteries, which can lead to a lot of trouble. Unsaturated fats, which tend to be healthier because they stay liquid at room temperature, contain double bonds. So with double bonds, which causes kinks, the kinks work like legs spreading out and pushing the neighboring triglyceride away from it. And so then the molecules continue to move, they don't solidify, and so then it's liquid at room temperature. Second category, of lipid is a phospholipid. So what a phospholipid has is a hydrophilic head, which means that it likes to bind to water. So philic for like. And then it has one to two hydrophobic fatty acid tails. What this allows is for us to produce plasma membranes or cell membranes. It's a cell membrane, and you get basically this interaction or two-layer. It's called a phospholipid bilayer, which we'll talk a little bit more about in the next lecture. Phospholipid bilayer, and this equals the plasma membrane. But it has a hydrophobic tail, hydrophilic head. The hydrophobic tails between two layers interact, so they stay together, and it basically creates a wall that separates inside of the cell from the outside of the cell. The last of lipid is cholesterol and its derivatives. Cholesterol is a hydrophobic molecule. And the cheat or easy way to see a hydrophobic molecule is something that really doesn't have any or very few hydroxyl groups. So this hydroxyl group will bind water or interacts with water, but the majority of the molecule has no hydroxyls or no oxygens, so it's hydrophobic. And how you identify a cholesterol or derivative, so a lot of the steroids in the body are cholesterol derivatives, is that they have four rings of carbons. With then side groups that basically differentiates the different type of cholesterol derivative. This is shot backwards with side chains. And some of the examples of the derivatives are the steroids of cholesterol, or the steroids that function as signal molecules, with probably one of the best examples, testosterone, or the female equivalent, or it's not really the female equivalent, but the other hormone that's typically thought to induce female characteristics, which is estradiol, which we will talk more about both of those later in the semester when we talk about the endocrine system. Okay, with that, we get to the proteins. So proteins are amino acids. So amino acid basically has an amino group on one end and a carboxyl group on the other end. They're called amino acids because this carboxyl group will actually donate the proton here, making it basically acidifying the solution it's in. So in our body, in almost all animals, we have 20 different amino acids that are found. They all have the same basic structure. So here you've got the backbone of an amino acid with the amino group and the carboxyl group, and then a side chain excuse me, that is what differentiates each of the amino acids. So the side chains, which you can see here, are these yellow or green or pink or blue, are what provide all the specific properties of an amino acid, and it's the equivalent of making amino acids into a variety of different Lego shapes. So polypeptides are amino acid chains that are made up of more than one amino acid. Joined together. And similar to carbohydrates, polypeptides are synthesized through dehydration synthesis, in which you basically have the hydroxyl group and a proton get released and a new bond formed down here, which is called a peptide bond. Not a peptide bond, peptide bond. So every polypeptide has a specific sequence of amino acids. So you can see here there's 3 different side chains with a directional amino group. So this is the N-terminus amino group and the carboxyl C-terminus. So giving the protein a orientation. Here this is methionine, tyrosine, and cysteine, sort of chained together in a very short polypeptide. The simplest level of protein organization is a sequence of amino acids in a polypeptide. So here you can see a very long chain, and just zooming in you can see phenylalanine, leucine, serine, and cysteine sort of in this one segment here. But as you get into these longer chains, you can see a high level of complexity starting to develop. The amino acid sequence is responsible for the structure, which is then confers the function of the protein. So confers function. So in this picture you can see an example of a couple of proteins. Proteins are polypeptide chains, either one polypeptide chain by itself, which you can see here is an example of poly-A polymerase, or ATP synthase, which is made up of multiple polypeptide chains, but is still considered a single protein. So single versus multiple. The function of the protein is dependent on its structure, so as long as it has the right structure, it will have its proper function. The proteins are some of the most varied of the biological macromolecules, giving them a lot of different functions. So some of the functions that proteins are able to do is maintain the structure of the cell. So structure, they're involved in generating all movement of the body. So muscle contraction is the interaction between proteins. They help stick cells together, so cell-cell adhesion. They help cells recognize each other as part of the immune response, and also just cells knowing that they're in the right location. Recognition. They're involved in transport, communication, and also work as enzymes, meaning that they help catalyze reactions. When a protein loses its structure, it's known as becoming denatured. So loss of structure. And loss of structure means loss of function. So denaturing a protein, typically when we cook something, that's denaturing a protein, and then it no longer is alive, no longer functions properly. So one category of protein are enzymes. And so here's two examples of enzymes again. The way that an enzyme works is that it helps catalyze reactions. So enzymes catalyze reactions, which is just another way of saying that they facilitate. And how they facilitate reactions is they reduce by reducing activation energy. So, and when that happens, then the reaction speeds up. So another way to look at this is a graph with time on the bottom, and here we have activation energy, or energy, so on the y-axis. So for those of you that have taken physics or chemistry, it's the delta G. So if you have a molecule with a certain amount of potential energy, there's then what's called as the activation barrier, that then once you get over it, the reaction occurs, then the molecule breaks apart and goes to, so this is to products at a lower activation energy. What an enzyme does is reduces this activation energy so that it takes less energy to get the reaction started, which functionally speeds up the reaction. A way to think of this is also, there's this illustration with frogs, where if you look at frogs as the reactants, frogs in the bottom pool is the products, the activation area energy is that barrier. What an enzyme would do is come in, remove some of those rocks, so now these hopping frogs, which are just hopping up and down, are more likely to get high enough to get over the wall, and so the overall reaction speeds up and you end up with more product. So the important thing to remember about enzymes is that they are not changed by the chemical reaction that they facilitate. So they're not really part of the reaction, they just assist in the reaction. Okay, last category is nucleic acids. So nucleic acids are single or double carbon nitrogen rings. So that's what we have here. So single or double carbon nitrogen ring. You have a monosaccharide, so a single sugar molecule. And then one or more phosphate groups. So here's the phosphate groups. So good place to start with this is a monomer form of a nucleotide, which is ATP. ATP, adenosine triphosphate, is the molecule functions kind of as a battery for the body or for our cells, basically storing energy by adding in a third phosphate here, which then it can then release through hydrolysis and produce energy that can be used for cells. ATPs are used, basically generated through the breakdown of glucose. And so as glucose gets oxidized, here you can see this is a whole cellular respiration process. So one glucose molecule yields approximately 36 ATP, depending on the cell that's doing it. This is something you will learn more about if you take biochemistry or introductory biology. So one of the primary other functions of nucleotides is that they are involved in encoding heritable information. So if you remember, the protein function is derived from its primary structure. Primary structure is just the sequence of amino acids. And so a sequence of every amino acid is encoded by a sequence of nucleotides, with there being two types of nucleotide. So there's deoxyribonucleic acid, or DNA, or ribonucleic acid. And the main thing I want you to take home from that is that DNA encodes the information. Basically, it works as a library, storing the information on how to make proteins. And so then DNA then becomes transcribed into RNA. So an RNA basically transports information to the ribosomes, which then translate the RNA sequence into proteins. And that's the end of this lecture. So next lecture, we'll be talking about membranes and cell biology. I will see everybody soon.